So let's study about a new gene interaction today, that is comb pattern in poultry. What is comb? You might have seen a red colored structure on the head of fowls. That is said to be comb. Mainly 4 kinds of combs are seen among fowls. First one is called pea comb, second one rose comb, another one is walnut comb, and the other one is single comb based on morphological variations. Why these variations occur regarding the comb pattern? It's due to 2 genes which are located in 2 different gene loci. Okay, so 2 genes located in 2 different loci are responsible for the comb pattern in poultry. So let's see how these genes interact to produce these 4 various phenotypic expressions. So I told you 2 genes are there. So I will explain with the help of chalk pieces. Suppose this is the dominant allele of the first gene, say R gene. So any alphabet can be used for representing gene. For convenience, we are using R for rose comb. So this is the dominant allele of R gene, and at least one dominant allele of R gene is required for the formation of rose comb. Now suppose this is the dominant allele of P gene. At least one dominant allele of P gene is required for P comb production. Now for the walnut phenotype formation, at least one dominant allele of both the genes are required. So capital R, minimum one capital R, one capital P. So both the genes should have their dominant alleles. Okay. So this is the condition that a walnut comb is produced. Now when the single comb is produced, when the dominant alleles of both the genes are absent, it leads to the formation of single comb. Okay. So what will be the genotype of single comb? Only one possible genotype is there for the single comb, that is small r, small p. This is the only possible genotype for single comb formation. Okay. So what are all the possible genotypes of walnut comb formation? There are 4 possible genotypes for walnut comb formation. See, this is one of the combinations. One of the possible genotypes, capital R, capital P. It is double dominant condition. Okay, homozygous and double dominant. Here both the alleles are dominant regarding R as well as P. So it is walnut, of course. Now the next condition is this one, capital R, capital P, small p. Another condition, capital R, capital P. Then one more condition is there, capital R, capital P, small p. Okay. So these are the genotypes, different possible genotypes. It is double heterozygous, you can see. So why is it a walnut? Because our condition is that at least one dominant allele of both the genes are required. Here one capital R is there, here one capital P is there. Okay, so it is walnut. Now regarding rose comb, what are all the possibilities? So for rose comb, it is one possibility, that is capital R, small p. Right? Another possibility, capital R, small p. Only 2 genotypes are available for the phenotypic expression rose comb. Similarly, for P comb, small r, capital P. This is one possibility. Next possible genotype is small r, capital P, small p. Got it? Now let's see what will be the F1 generation produced when a rose comb organism is crossed with a P-combed one. Okay. So, a pure rose-comb, that is capital R, small p, when it is crossed with a pure P-comb one. See, this is rose-comb, it is P-comb. Okay. So, how many gametes can be produced from this individual? You all know only one kind of gamete is produced. This is a gamete that can be produced from this organism. Capital R, small p. What about this organism? Here also, this is the genotype of the second individual, second parent. Here also only one gamete can be produced, small r, capital P. Okay. So, this is the gamete of the second parent, this is the gamete of the first parent. Now, what is the genotype of F1 individual? Capital R, small r, capital P, small p. So, this is the genotype of F1 organism. So, what will be the phenotype? It is walnut, because one capital R and one capital P are present. So, dominant allele of P gene, R gene, dominant allele of P gene are present. So, walnut comb is a phenotypic expression. Hope you understood. Okay. Now, let us see which are all the gametes which are produced from this. It is very clear, no? So, this can fuse with either this or this. This can fuse with either this or this. So, 4 kinds of gametes can be produced: capital R, capital P, capital R, small r, capital P, small r, small p. Now, let us see what will be the offspring which are produced in F2 generation, and let us find out the F2 phenotypic ratio also. Okay. So, we are talking about comb pattern in poultry or fowls. We crossed a rose comb parent with the P comb one. Both are pure for its character. You can see the genotype of rose comb parent, capital R, small p, and that of P comb is small r, capital P. You can see the gametes formed from both the parents, and the F1 genotype is also shown there. So, these are the different gametes which can be produced, which was produced from F1 organism. And see, this is the Punnett square that shows the F2 offspring. See, you count the number of walnut combed organisms or walnut combed fowls among the F2 offspring. So, among the F2 offspring, 1, 2, 3, 4, 5, 6, 7, 8 and 9. So, 9 walnut combed offspring are seen to be produced, right? Now, how many rose combed ones are produced? 3. Likewise, how many P combed ones are produced? 1, 2, 3. So, 9 walnut comb, 3 rose comb, 3 P comb, and of course, this is a single comb, only one possible genotype, and here you can see 1 here. Okay. So, the phenotypic ratio of comb pattern in poultry is 9:3:3:1. It is similar to the dihybrid F2 Mendelian ratio. Okay. So, that's all about comb pattern in poultry. So, once again, it is controlled by 2 genes which are located in 2 different loci. How these genes interact? The dominant allele of R gene is responsible for rose comb formation. Dominant allele of P gene is responsible for P comb formation. At least one dominant allele of both the genes, when present together, it can lead to walnut formation. And when the dominant allele of both the genes are absent, that leads to single comb formation. and you have seen the F2 ratio also. It is 9:3:3:1, similar to the F2 dihybrid Mendelian ratio. Okay, hope you understood.